Students, today we'll be studying the last two deviations, that is multiple alleles and pleiotropy, from our third lesson, that is inheritance and variation. So first we'll study about multiple alleles. Multiple alleles can be defined as when more than two alternative forms or alleles of a gene occupying same position or locus on a chromosome or its homologue is called as multiple alleles. The word multiple means many. Hence the name multiple alleles, because till now we have studied that a gene has only two alleles, but in multiple alleles a gene will have more than two alleles. Hence the name multiple alleles. Multiple alleles arise by mutations. A single gene can mutate several times and produce alternative or different expressions. The best example to study multiple alleles is Drosophila. Drosophila, it is an insect which is also called as fruit fly. Now in Drosophila, large number of multiple alleles can be seen. Some of them are the wings of Drosophila. So this is the example that we'll be studying. The wings of Drosophila, it shows alternative expressions. Now what are the alternative expressions? The wings of Drosophila, it ranges from normal wings, which is a wild type, to no wings, which is also called as vestigial wings. What are the different forms of wings that can be seen in a Drosophila? We'll see it with the help of a table. Now in this table we can see the different phenotypes as well as the genotypes for the wings of Drosophila. The first type is normal wings. This insect or this Drosophila, it will have normal wings and it is denoted by VG+. This is the genotype. The next one is nicked wings. Now the wings may have a cut and it is denoted by VGNI. NI, the first two letters, hence VGNI. Then comes the notched wings. The wings may show a notch and denoted by VGNO. The first two letters NO. Then comes strap wings. The wings may appear like a belt or a strap, hence the name strap wings, and it is denoted by VGST. Then the last one, vestigial wings, that is the insect will not have wings at all and it is denoted by VG. So this is the dominant one, and vestigial, this is the recessive trait. But intermediate characters or traits can also be seen. This was the first example. We can study one more example, and the second example is ABO blood group in human beings. Now this blood group, it is controlled by gene I, and this gene I, it shows different alleles such as IA, IB and small I. These are the different alleles, and these alleles, they show or they represent a particular blood group. Now since human beings are diploid, a human being may contain any two genes or alleles and will represent the blood group related to that. For example, if the genotype of the person is IA, this is the first genotype, or if the genotype is IA and small I, then both the genotypes, they will represent the blood group as A. For next genotype, if the genotype of the person is IB, or I, B, I, still the blood group of the person will be B. The third one is if the genotype of the person is IA and IB, then it will represent the blood group as AB. But if the genotype of the person is small I, then the blood group of the person will be O. So this is the phenotype of the blood group and this is the genotype of the blood group. For 4 blood groups, there are 6 genotypes. 1, 2, 3, 4, 5 and 6. Out of which this is dominant and this is recessive. So these are the genotypes which will represent the phenotype or the blood group of an individual. So these 2 examples were included in multiple alleles, that is Drosophila or wings of Drosophila, and blood group in human beings. Coming to the last deviation, that is Pleiotropy or pleiotropism. When a single gene controls 2 or more traits, then that gene is called as pleiotropic gene and the phenomenon is called as pleiotropy or pleiotropism. The example of pleiotropy is sickle cell anemia. The word anemia, it is related to blood. So sickle cell anemia is a disorder of blood in which the RBCs become half moon or sickle shaped in low or oxygen deficiency. HbS is the gene which causes the disease, that is sickle cell anemia, whereas HbA is a normal gene. These are the phenotypes and genotypes for sickle cell anemia. The normal or healthy person will show genotype as HbA, homozygous condition. The carrier person for sickle cell will show genotype as HbA, HbS, heterozygous. And the person suffering from sickle cell anemia, it will show HbS, HbS as its genotype. So when this gene is present in double dose, it is very dangerous or lethal and it causes death of the person. This is dominant gene and this is a recessive gene. So if the person is having recessive gene in double dose, the person will suffer from sickle cell anemia and it may lead to death of the person. So this is about the phenotype and genotype. Now let's study pleiotropy or its example, that is sickle cell anemia, with the help of graphic representation of pleiotropy. So this is the graphic or diagrammatic representation where we have to perform or do a cross between the parents. So parents, their phenotype is both the parents are sickle cell carrier. So when we perform a cross between the parents who are sickle cell carrier, this will be its phenotype. Its genotype will be, as we have seen just now, it will be heterozygous. So it will become HbA and HbS. Similarly, for other parent also, it will become HbA, HbS. So these are the genotypes. Then comes the gamete formation. So the gametes will be, since it is heterozygous, it will produce 2 different types of gametes. One will be HbA, the other gamete will be HbS. These are the gametes for the first parents. The gametes for other parents will also be same. It will also be like HbA and HbS. These are the gametes for male parent and female parent. Now, once the gametes are formed, the next process is of fertilization. So what we have to do? We have to fertilize one gamete from male parent and the other gamete from the female parent. So first we will take this gamete and we'll cross it with the other one. So what will be the result of that? If we make it like this, this is the first one, it will be HBA. Now we have to cross it with the other one, so it will become, it will be HBA. When both are HBA, HBA, it will be, the phenotype will be this offspring, it will be normal. Similarly, we have to do the cross for the other one also. So now we'll take this same gamete, but this time we'll cross it with the other gamete from the female parent. So this is the female parent. Now it will be, what will be its genotype? One is HBA. So we'll write it here as HBA. What is the next one? HBS. So HBA and HBS, these are heterozygotes. So this offspring, it will be sickle cell carrier. So this is sickle cell carrier offspring. Now coming to the next one. So now this gamete is remaining. This we have to cross once with this and next with this one. So it will be like this. First we'll take this one. So then it will be, it will come here. And now we have to cross it with this one. So it will be this gamete and this gamete. Its genotype will be, what is the genotype of this? HBS. This is HBA, but still we have to write HBA first and then we have to write down HBS. So it will become HBA and HBS. HBA, HBS both are same, so its phenotype will be same. It will also be sickle cell carrier. So there are two offsprings which are sickle cell carrier. Now the last one is remaining. We have to cross this gamete with this one. So we'll do a cross between them. This is one gamete. We have to cross it with the other one. This is another one. Now we have to mention its genotype and phenotype. What is the genotype of this? HBS. And this also HBS. So it will be HBS, HBS. Since both are recessive, the offspring will be sickle cell anemia. It will suffer from the disease which is called as sickle cell anemia. So these are the genotypes and these are the phenotypes of the offsprings that are produced. Now there are 4 offsprings that are produced. The first offspring is normal, the second and third offspring is carrier, and the last one is having the disease, that is sickle cell anemia. Now we have to calculate the ratio. So the ratio will become 1:2:1. So 1:2:1. 1 represents the normal offspring, 2 represents the carriers, and the last one represents the sickle cell anemia. But as we have already said, when the offsprings or the individual are carriers, then they will show mild anemic symptoms and they will carry the traits for that disease. But sickle cell anemic person, when it carries recessive genes in double dose, it becomes lethal and it causes the death of the individual. So what happens when the ratio is 1:2:1? The normal, it does not have any problem. Carriers, they carry the traits, they carry the genotypes, but they will just show signs of mild anemia, whereas the last one, it dies. So finally, the phenotypic ratio, it becomes 1:2. This is the final phenotypic ratio because the last offspring, it dies due to the disease, that is sickle cell anemia. Hence the ratio becomes 1:2. So this you have to remember for your exams. Now the basic difference between pleiotropy and multiple allele is, in multiple alleles, the single gene may have more than 2 alleles, whereas in pleiotropy, a single gene controls 2 or more traits. This is the difference between multiple alleles and pleiotropy. So this is all about the deviations. Thank you.